Now invite Dr. Hina Bansal. Dr. Hina Bansal.
Yes, sir, I'm there. Just give me a moment. I'm sharing my presentation. So myself, Dr. Hina Bansal, I am Assistant Professor in Amity University, Noida, and I would like to thank the organizer to give me the opportunity to present my work entitled Unveiling Therapeutic Opportunities, Subtractive Genome Analysis of Providencia Stuartii for Drug Target Identification and Epitope Mapping. As Providencia stuartii is a gram-negative pathogenic bacterium associated with various types of infections in human, specifically in immunocompromising situations such as urinary tract infection, wound infection, respiratory tract infection, and blood infection. And it is known for its multi-drug resistance. So the current research used a subtractive genome analysis that approach to identify essential enzymes that are unique to the pathogen and can be used as a potential drug target against its pathogenesis. The subtractive genome analysis is a powerful approach in genomics, enabling researchers to focus on specific genetic differences that may have significant biological implications. It will provide a foundation for developing new therapeutic strategies against the pathogen, which poses significant challenges due to its resistance to multiple antibiotics. So the research objectives of the current study are to enhance the understanding of Providencia stuartii and uncover novel therapeutic avenues for addressing infections caused by this opportunist pathogen, and can be outlined as identify unique genetic elements. It is by utilizing subtractive genome analysis, one can pinpoint genes and genomic regions that are unique to Providencia stuartii, especially those related to pathogenicity and antibiotic resistance. Next is characteristic potential drug target. This is the analyze the unique genes identified for each potential as drug target, focusing on essential genes that are conserved across related pathogens but absent in the host. Then evaluate functional roles. It is the investigation of the function to identify genes through in vitro and in vivo studies to assess their role in the pathogenicity of the pathogen. Then epitope mapping to identify and characterize epitopes derived from the unique protein, facilitating the development of vaccines or diagnostics tools. Then develop therapeutic strategies based on the findings. Propose novel therapeutic strategies, including potential drug formulation and vaccine candidates, aim at combating infections caused by Providencia stuartii. So this is the methodology that can explain that we first extracted the metabolic pathways of the human and the pathogen from the CAC pathway database. Then manual comparison was done, and the pathways that are present only in pathogen will be considered as a unique pathogenic pathways, and the common pathways will be removed from the study. Then all the enzymes were extracted that are present in the unique pathway using the GapTop tool. The essentiality of those genes have been identified. Those genes which are shown essential to the pathogen are included in the study, and non-essential genes are eliminated from the study. Further, all the essential genes were subject to blast homology search and found homologous to the human host will be discarded, and only non-homologous protein which are not presenting any similarity with the human genome, these are included in the study. Then the PPI network was constructed using the string database. Then all the PPI network constructed for all the non-homologous essential proteins, and M-code analysis was conducted to find the hub genes, the cluster genes that are mostly related and having the highest rank in the highest cluster module. Then subcellular localization was identified, followed by the toxicity analysis. Then gene ontology analysis was performed, and finally the drugability testing was performed. All the Genes which are having existing drugs means the hits found will be discarded, and all the non-hit found proteins, their drugs are not found, selected as a novel drug targets. Then finally, the epitope mapping was conducted and find using the ABC-PRED server. Then moving on to the result section, we find the KEC pathway database was employed to extract the metabolic pathway of the pathogen and the host. A manual screening was done to discover the pathway exclusive to the Providencia Stuartii. Metabolic pathways that were exclusively present in the pathogen were considered as unique pathway and assumed to be responsible for showing the pathogenicity in the host. A total of 93 metabolic pathways were extracted, out of which 28 were found to be novel to Providencia Stuartii after manual comparison with host metabolic pathways. All the enzymes involved in these 28 distinct pathways were extracted and screened for their essentiality. A total of 298 enzymes were predicted to be essential for the pathogen and identified as non-homologous, as they did not show any homology with the human. Now, these 298 non-host essential proteins were subjected to a string database and a PPI network was obtained. The most likely metabolic functional relationship between these selected proteins were determined by looking at the highest rank cluster module having 25 nodes. So these are highlighted in yellow color in the network. A protein-protein interaction network represents the physical and functional interaction between protein within a biological system. Understanding these networks is crucial for elucidating cellular processes and disease mechanism. The selected higher rank module, which is having 25 protein, was subject to gene ontology analysis. Here you can see in the gene ontology analysis, we can find that in biological process, 30% proteins are involved in lipid A biosynthesis, 25% proteins are involved in peptidoglycan biosynthesis, 23% proteins are involved in liposaccharide biosynthesis, and only 22% proteins are included in cell division. When we see the molecular function, then 48% of the proteins are APT binding, 25% are involved in transferase activity, 15% proteins are involved in kinase activity, and 12% proteins are involved in carbohydrate derivative binding activities. When looking for the cellular component, then we see most of the proteins are cytoplasmic. At 88% proteins are present in cytoplasm. Then 8% of the proteins are membrane protein and 4% are the other proteins. Now we check the druggability and toxicity testing. A novel drug target identification was performed using the drug bank database. Proteins that displayed no matching hits against the standard cutoff value were proposed as novel drug targets. Toxicity analysis revealed that none of the proteins were toxic in the nature, and the result revealed these 15 proteins as novel drug target, as they were specifically involved in unique pathways of the pathogen. These are listed in the table. And followed by beta cell epitope mapping. So beta cell epitope mapping was done by artificial neural network ABC-PRED program, which indicating these 3 proteins which are present in the membrane and having the score greater than 0.9 considered as a vaccine candidate. Now we can summarize our work in this figure only, which is showing a total of 1084 protein. We find 358 as an essential protein, out of which 298 are non-homologous protein, in which 25 are having functional association, and out of which 15 can be suited as the best novel drug targets, and 3 proteins as a subtractive genomics. So the identification of the key findings can be the identification of several potentially drug targets that are unique to the pathogen and are not present in human. These targets could be important for developing new antibiotics or therapeutic agents. Then identifying these unique drug targets help develop antibiotics that specifically target pathogen without affecting the host. Epitopes, specific epitopes were identified that could be used to design vaccines or diagnostic tools. And this can aid in creating vaccines that elicited the strong immune response against the pathogen. Now, when we are moving to the conclusion, so in conclusion, we can summarize that the study aimed to address critical gaps in our understanding of this opportunistic pathogen. By leveraging subtractive genome analysis, we have identified unique genetic elements that could serve as potential drug targets and epitopes for vaccine development. The finding of this research not only contributes to the fundamental knowledge of Providencia Stuartii, but also hold promise for developing novel therapeutic strategies to combat infections caused by this organism. Further, experimental validation of the identified targets and epitopes is needed to confirm their efficacy and safety. Future research can build upon the foundational work laid out in this study, ultimately leading to effective intervention against Providencia Stuartii and enhancing our ability to combat opportunistic infection more broadly. Thanks for your attention.
Thank you, Dr. Hima, for the wonderful talk on the bioinformatics session, particularly on epitopes and also subtractive genome analysis. Very interesting. Really, I felt quite happy when you were mentioning about the epitope mapping and all that. But one thing, why did you not test it with HLA2 mapping? Directly, you were telling B cell mapping.
Yes, sir.
HLA2 binding, you have to, so for all bacterial proteins, you have to bind, you have to see the binding potential to HLA2, no?
Sorry, sir, your voice is not clear to me.
No,
Can you repeat your question?
the binding potential of the epitope sequence to that of the HLA2 is almost mandatory to evaluate its epitope potential, no?
Yes, sir. Yes, sir. This is our future. This work is still going on, sir. And we will definitely consider that point which you have suggested. And we'll definitely go for it. We will check the binding affinity. Yes, sir.
Yeah, it is sequential epitope, madam, or is it, there are epitopes which are sequential, there is some other epitopes, no? Is it sequential
Yes, sir.
epitope? Yeah.
Yes, sir. These are sequential episodes.
Allergic test did you do? Toxic
No,
test
sir,
you have done?
that still we haven't done. So we will moving forward for that part also.
Yeah. So you have come down to 28 novel metabolic pathways.
Yes,
That
sir.
includes even even shikimate pathway.
Sir,
In
these
bacteria,
pathways are, sir, I can show you the list of the pathways. It is there with my supplementary file. If you want to see the list of the pathways, what are the basic pathways? So I
the
can, yes,
no
sir.
problem.
I
Shikimate
can
pathway is very popular
share.
among the shikimate. Okay, no problem. It's
Sorry,
good
sir,
talk.
I'm not
Thanks.
getting your point.
Shikimate pathway is popular among
Shikimate
the
pathways.
that is popular
Okay.
among bacteria, but not in humans. So all the
Okay.
drugs
Okay.
are being directed against the shikimate path.
Yes, sir. Yes, sir.
That's what I'm asking, whether this shikimate pathway, does it include out of the 28?
Yes, sir.
What you
Yes,
are
sir.
mentioning?
Yes, sir. Yes, sir.
Yeah, that
These are
also.
included, sir.
Thank you, ma'am. Thank you for the wonderful talk.
Thank you, sir.